So first of all, I would like to start my presentation in drug discovery value chain in this slide. So as you know very well, drug discovery is consistent the several stage described here. Actually, it is focused until to the IND enabling study. So we have the 6 stage actually. So we have the multifunction is the generating to the knockout TG mouse and the in vitro cell-based assay and PK/PD analysis. It's combination with the modeling and the simulation studies. Also, we have the disease model using to the knockout transgenic mice and the biomarker also. So today, I'd like to focus on the 2 early stage. So our big question is how to identify and validate potential drug target, which is when looking into the treatment for the specific disease. So as you know very well, the target identification is the first step in the discovery a new drug. So important thing is to identify the function of the positive therapeutic target. So another thing is that identify the molecular mechanism is the disease, which would have a significant impact on the success rate clinical stages. So on the other hand, the target validation, that one is the prioritization of the drug target candidate from the target identification described below. And the biggest aim is to get the preclinical POC with therapeutic effect. That means to the molecular target is involved in the disease process, and the modification of the target would have a therapeutic effect. So this slide shows the success rate of the clinical stages too. The reference is the Nature Review Drug Discovery 2016. It's a little bit old one, but I still this data are effect on the success rate one. So as you can see here, the main reason for the fail is the efficacy. I mean, to the lack of efficacy. So in phase 2 and phase 3 is same result. Efficacy is very important. Another thing is the commercial and strategic decision is the 20% of the reason for the fail in clinical stage. So this information indicates that the 2 points of the success rate on the clinical stage. So one is the increase of the efficacy is the key success factor in clinical stage. So second is the 20% of the asset have the still potential. So again, so the approach for the 2 point described to the previous slide. Number one is the lack of the efficacy. I mean, to the how increase to the efficacy in the clinical stage. So I think they have the 3 point. The one is the target identification. That is the topic in my presentation would have the significant impact in drug discovery research. So second part is the second one is for the clinical POC. Multiple validation approach will be the best way. So last one is the bridging between the preclinical and clinical. So suitable biomarker will be important. So second, due to the strategic decision, so aspect from the maximize the asset. So repositioning approach will be impact. So today, I would like to focus on the number one topic. So, and this slide is a little bit complicated, but I would like to introduce you the Axcelead Drug Discovery Engine, which is one of the concepts for the target target identification and validation. We could find the new target and validation through this engine. So starting point is the direction for the drug generation. For example, maybe the client have the strategy such as therapeutic area, or maybe a client have a focused pathway or focused gene on the genome. So we received some data from client and demonstrate to the omics analysis using to the transcriptome or proteome or metabolome and lipidomics. So data from the client is included into the cell line data or animal data or clinical data. So after that, receive to the some data, we make to the drug candidate list from using to the enrichment analysis or network analysis, or we can use to the Axcelead original fingerprinting analysis. And additionally, we can approach using to the development status using the Cortelis. So that one is the target identification. So after that, I mean to the find to the candidate list for the target. We to the target validation analysis using to the in vivo model and prioritization of the drug target candidate. So again, so target identification means the identification of the molecular target described on previous slide. And in addition to the molecular target, asset identification could be crucial for the drug discovery stage. So actually, Axcelead has the two approaches, such as phenotypic drug discovery and target-based drug discoveries. So I want to focus on the asset identify the strategy and the story next slide. So we have the two approaches toward the novel small molecule targets. So as mentioned the before slide, we received to the direction from the client according to the drug generation. So we have the two approaches described here. One is the phenotypic oriented, so another one is the target related. So both of approach have the pros and cons. For example, phenotypic oriented, I mean to the phenotypic drug discovery, is the not need to know the molecule target as disease of disease, and that's one possible to discover the some compound with unique MOA, but this is low throughput. On the other hand, target-based discovery is high throughput, and we can approach use to the structure-based approach as possible. So I think we more focus on the phenotypic oriented, but the most cons point is target deconvolution after the phenotypic screening. So for the solution for the this point, we have the target deconvolution method. And actually, we have the 2 points. One is the chemical approaches. Another one is the compound fingerprinting approach using the transcript analysis. And this slide, I would like to touch on a little bit to the fingerprinting approaches. That one is based on the transcriptome data using the RNA sequence. So we can treat it some cells with a compound, and we get to the transcript data from the cells. And we will try to find to the target from the fingerprinting data. So this one is some expression data from the treated cells. Then we try to compare with the published database. That one is the expression data, fingerprinting data. So after that, we select to the more similar compound, so that we can speculate to the MOA or some molecular target. So success rate is around to the 70% on the top 3 between the top 10. So another approach is the chemical proteomics approaches. We have two methods. One is the PAL, photo affinity labeling approaches. So another one is the ABPP approaches, activity-based protein profiling. So that one is very powerful because we can do the direct target fishing. I mean to the direct binding of the compound with the protein with using to the compound. So that compound have the efficacy on some cell line or in vivo variation. So another point is that we can use the early stage compound such as the lead generation or optimization. So that two approaches, chemical proteome approaches and the fingerprinting approach, we can strongly recommend it to the phenotypic screening to find to the small molecule. So I want to little bit change to the topic for the target validation. So as you know very well, so target validation consists of the in vitro, in vivo, and biomarker. So for the in vitro validation, we have some platform that is including to the cell line from the human cells, which is collaborated with the HAB, HAB organization, and the IPAC facility, which is located in Japan, Axcelead. So also it was available IPS drive sample, which is collaboration with the Fujifilm. For the in vivo validation, we could cover the 6 therapeutic areas described here. So each therapeutic area has a specialist who has the strong background and expertise through the Takeda pharmaceutical company. So in next 3 slides, I would like to show you some example about the in vivo validation study. So therapeutic area is the kidney disease. We have original chronic kidney disease model. That one is a mouse. So we call to the AXCC mouse, AXCC mouse. That is the full spell for the name. So this mouse is deficient for the collagen type 4A mutation. Sorry, it's a mutation mouse. It's not deficient mouse. So as you can see here, the staining of the collagen type 4, that one is the wild type mouse. That one is the mutation mouse. So you can see here the staining of the collagen type 4 is gone. So in this slide, I would like to show you some in vivo validation study using the Axcelead mouse. So left side is the this study. So that mouse is eventually die due to the kidney failure. That's black line is the Axi mouse. Around to the 30 weeks, 60% of the mouse is dying. So and historical evaluation, so abnormal glomerular-based membrane is observed. Infiltration of the inflammation cells is observed. Fibrosis is observed, and loss of the food process in the podocyte. So this phenomenon is typical for the chronic kidney disease. So in using to the Axi mouse, we try to the validation for the some drug. That one is the angiotensin 2 receptor blocker, erisperosan. So red one is the drug treatment. As you can see here, after the 80 weeks, something, we can decrease to the urine albumin expression. So according to the in experiment, MOA, mechanism of action analysis will be very, very powerful to understanding the efficacy in model mouse. So Axcelead have the several approaches, including to the spatial transcript analysis. That one is the hybrid to the histological analysis with the expression data. Another one is the histological evaluation, as described before. Another one is the laser microdigestion and single cell and nucleus RNA sequence analysis. So using these approaches, we relate to the MOA from organ to the single cells level. And this slide, I would like to touch on a little bit to the biomarker. So we actually use into the several material like blood sample, organ biopsy sample. So we use to the omics analysis. We can find to the candidate for the novel biomarker. So after finding to the candidate for the novel biomarker, we try to evaluate these biomarker between the disease progress, and biomarker changes. So finally, we could bridge in clinical development and preclinical data stage using modeling and simulations. So I would like to change the topic a little bit. So in previous slide, I presented the typical example for the in vivo validation, but I sometimes to take more aggressive approach more quickly. So one of the answer is the gene modification. So as you know, gene modification is the symbol for the drug discovery, but the current gene modifier is the needed model. So our solution is the transientry gene expression animal model using hydrodynamics gene delivery, HDD. So this is one of the feasibility study of the HGD using luciferase transfection. As you can see here, the left panel, luciferase signal is observed in day 1 through the day 7. In middle panel, repeated dosing result. So you can see here, so this model is the repeated dosing is applicable. On the other hand, some toxicity is observed, but after the 14 days is graver, toxicity on liver is recovered. I think this result indicates that the toxicity is manageable. So in my last slide, I'd like to touch on our company a little bit. So we are a solution provider in drug discovery space from Japan. So established in 2017 as a Takeda spinning out company. And the number of employees around to the 300. So location is the Shonan Park. So we have a very strong scientific leaders and cover to the capability from the small molecule and the peptide early stage to the optimization for the clinical development. Also, we have some strong partnership with the new technology and the Japanese government relationships. And so thank you all very much.